Okay, folks, what's up? We're going to address myth number 2 on lactic acid. So it's this one. Lactic acid is a waste product. And when I say waste product, what I want you to think is predominantly or exclusively a waste product. You're gonna find lots of things in urine and feces, right? Like lots of things, vitamins, minerals, right? And so if we were going to define lactic acid as a waste product, because let's say we do find small amounts of lactate in our urine and in healthy individuals, right? Like, but in a small percentage as well, as far as the amount of lactate that we make, right? And so if we were going to, we would define so many minerals and so many vitamins also as waste products, right? So yeah, do you find some in your urine? Absolutely, minimal amount in healthy individuals, right? But so let's look at some things, right, that we actually use it into. So first one is your inactive skeletal muscles, right? Now, all muscles are relatively active at all times, right? They're doing something. Maybe they're balancing you, right? They're coordinating you, things like that maybe even you're not even aware of, right? Keeping you in positions and holding you and stuff like that, though you don't think you're creating massive amounts of force. So when I use inactive, right, let's not get caught up in that. But when I say inactive, what I mean is like you're not necessarily using them for an exercise that you're doing. So an example of what I would call an inactive skeletal muscle would be like, so if we were bicycling, right, some kind of thing like that, road cycling, I would say like your upper body. So anything, your arms, shoulders, something like that. Right? Those are going to be the things that I would say inactive skeletal muscle. So what happens is when your legs are going to be creating it, right, it would be dumped into the blood via these things called monocarboxylate transporters or MCTs, much on those later. Dumped into the blood, carried up, go to the arms, right? The arms could take it in, biceps, triceps, deltoids, right? Front, back of the arm, shoulders, things like that, right? And they could use it for fuel because in those areas, they're going to have sufficient oxygen in order to kind of carry out those processes of pyruvate oxidation and getting things into the mitochondria, things like that. So more on that later. We're a long way from that, but right within the mitochondria is going to create a lot of ATP, something that's oxygen is going to be needed for. So in the heart, right, the heart has a lot of oxygen available to it, sufficient oxygen in order that it can use lactate. That lactate can be turned back into pyruvate. It can be oxidized in the mitochondria and then made into ATP via the electron transport chain, oxidative phosphorylation, all that stuff. But the heart, the liver, a lot's going to go to the liver. The liver's also going to turn it into glucose via something we call gluconeogenesis, or the creation of new glucose. Something we'll talk about a lot later. That's called the Cori cycle, right? So it's something that we'll see, right? A useful thing. And in smaller amounts, right, you're going to use some in the brain, you're going to use some in the kidneys. And one of the coolest things I found out that to me shows us how amazing and complex we really are, is that you're actually going to use some of it within the same muscle that creates it, even though you don't think you would. But you've got two, we call them, let's say, families of fibers. What we call kind of like a type 1 fiber and a type 2 fiber. Now, I'll kind of break type 2 fibers down more a little bit later into type 2A and type 2X, let you know the differences. But in general now, let's just say type 1 fibers are more what we call aerobic, oxygen using. They'll have a large blood supply. They use a lot of oxygen. They need a lot of oxygen. They'll have large amounts of mitochondria. They'll have big mitochondria. They'll be very what I call mitochondrial dense. Mitochondria will take up a lot of space within that cell, right? Like that's how they'll make the vast majority of their ATP, their energy that they're going to use, what I call their energy currency that they'll use, right? Now, type 2 fibers in general, type 2X more so than A, but we'll talk about that later, but are more what we call anaerobic, meaning they're going to create their ATP, like for your movement. We're talking about skeletal muscle fibers, right? Skeletal muscle fibers, skeletal muscles in general. Type 2 fibers in general are going to be more anaerobic. So they're going to use things like phosphocreatine, right? So when you're going to take your creatine, right, that's typically what you're taking it for. so that you're going to kind of give yourself like larger than, let's say, stores of it than you could create without those kind of supplements within the muscles, and you could use that creatine to recover more quickly, be more, do more sets, more volume, et cetera, et cetera, et cetera, gain more mass via hypertrophy, or those kind of things like that, and glucose, right? So it's going to be what we call kind of a glycolytic. Though type 1 muscle fibers can use glucose just as well. They're not as reliant on it. They can use other things, but they can use it too. But type 2 tend to be more exclusively glycolytic. Type 2A kind of live in both worlds, right? That's what I like to say, but we'll get more on them. But they're still more anaerobic, right? They can still do more of their work. They're still more, and they're going to be more explosive, things like that as far as skeletal muscle fibers go. So these are all kind of reasons why I don't think lactic acid is a waste product, right? Because it's used in all of these things. Now, what I'm going to do is flip the board, and we're going to look at some of the other reasons that I kind of put on that paperwork. If you don't have the paperwork, that I'll show you right here. What's up, folks? So we're going to look at part B of why lactate is not a waste product, right? In kind of our Lactate Myth series. So, right, one of the things, the primary thing that lactate is associated with is glycolysis, right? So in glycolysis, right, just a word, don't be intimidated by it, right? When you see this kind of OLYSIS in words that we're going to be using, we're going to say glycogenolysis, proteolysis, lipolysis, things like that. When you see this, right, just think about like the breaking down of, the degradation of, right, the separation of, the splitting of, right? Because really what we're going to think about with glucose is we're going to split glucose, a 6-carbon molecule, that's not all of this, but right, a 6-carbon molecule into 2 3-carbon molecules, and then we're going to further break that down. Into acetyl-CoA. We've got to oxidize it. That's a whole other thing. We're not going to get into that today. But, right, so when we see this word, right, that's what it means. And so what that means is, right, this GLYC refers to the glucose. So we're taking a glucose and we are splitting it, right? And that process is going to net us some ATP. It's not a lot of ATP, but it's a relatively rapid, a relatively fast way to get ATP, right? It's not as fast as the system we'll talk about at some point, called our ATP-PCR system, or our ATP-CP system, phosphocreatine system, something like that, right? You might have heard those. And it's really probably why, if you supplement with creatine monohydrate, that's why you probably do it, right? For that system, to enhance that system's ability to do work. Whole other lesson on that. But, right, so it's going to help keep glycolysis running, right? The splitting of glucose, right? So it's a really important system that we use to kind of propel ourselves through space-time, as it were, right? So, right, you're going to take glycogen and it's going to become glucose probably, right, at some point. And if you're going to make energy with it, it's going to. So, right. Now, we're not going to go into glycolysis. It's a complicated process. I'm going to say it's not a complicated process. It's a complicated process. There's a lot of enzymes involved. And not only things, there's a lot of things that influence those enzymes, right? Cofactors, temperatures, pH, et cetera, et cetera, et cetera, right? And it's something I'll go into in depth, right? But for now, what you're going to have to just kind of accept is this very rudimentary drawing, right? And know that glucose, through a series of steps, right, is going to become 2-pyruvate, right? Now, this is the kind of important part that we're going to look at kind of here in group. Now, glucose to 2-pyruvate. Then those 2-pyruvate are going to become 2-lactate. Now, what you can see in green over here is, right, well, I was like, why I say you need lactate to keep glycolysis running. So, right, over here, right, in this process, right, what we're going to start doing is actually what's called oxidizing, right? So it's going to be a series of what we call redox reactions, and so that's reduction and oxidation reactions, right? And so with reducing, you're kind of adding electrons to something, right? And with oxidizing, you're removing electrons from something. Anyway, so things are always a series of redox reactions, right? Like if something's getting reduced, something's getting oxidized, etc. Right? But I don't want to get caught up in that. It's something once again we'll talk about when we kind of start talking about like learning the language and processes and things like that. But you've got this compound. It's called NAD+, right? You may have heard about it. People are starting to supplement with it and do various things with it because it's a very important process in so many metabolic processes that we'll talk about, that you'll see. But so it's called nicotinamide adenine dinucleotide, right? And biochemists and maybe biologists, etc. too, right, like me, are inherently lazy and we don't want to say that all the time. So what we do, right, is we just call it NAD. So things like phosphofructokinase, we call it PFK, right? So, right, nicotinamide adenine dinucleotide. Anyway, so I'll just call it NAD, but that's what it is. So you've got two of these that are going to be involved in this process, right? And so those two, what they're going to each do is it's going to remove 2 protons and 2 electrons from this process. Now, you don't see those, right? Because this H that you see right here, when it came off, it was a hydride ion. It was an H minus. It was a proton with 2 electrons. We're not going to get caught up in that. But then you've also got this H plus. Now, inherently, if you want to think about what pH is, right? pH is just a measurement. And when we talk about pH, acidity, right? As pH goes down, things become more acidic. As that scale kind of goes up, things become more basic, right? There's less, right? As the pH goes down, right, there's more and more free protons. That means that environment is what we call more and more acidic. That's the term we've given to that, right? That process, right? So by the nature, right, of breaking down glucose to 2-pyruvate, this is something I'm going to allude to later. I know it doesn't seem super cool now, but it's very cool, right? I'll allude to this later when we look at how lactate doesn't actually, or the myth that lactate is what causes the rise in acidity, and I'll say it's actually not. But this is something I'll allude to, right? That 2 NADH and an H plus are going to get released into the environment, right? So they're released into the environment and they're floating around, right, in this kind of milieu, this kind of soup or whatever, your cytoplasm, your sarcoplasm, more appropriately, your sarcoplasm, right? All right, so now what's going to happen is, right, for this process to continue. So you have these 2 NADH plus H pluses. Here's your problem, right? This is why lactate is so necessary for this process. For glycolysis to continue running, right, for it to keep going, for the, if you're going to maximally exercise, let's say up to 1 minute of exercise, for it to be the predominant force of creating ATP, right, you're going to need to make sure you have an abundance of NAD+, right? So what you've got to do is what I call recycle it, right? You've got to flip it. Now, you can't do that really quickly without lactate. There is a way to do that. To turn NADH and H plus back into NAD plus. There's a way to do that. But it involves, right, the mitochondria, right? It's a much slower process, many more steps, et cetera, right? This needs to be rapid, right? Rapid. That way of recycling, for lack of a better term, your NAD plus is too slow for you to keep up that rate of ATP creation. Right? Which is what you need. That's why you're using that system, because you needed to be explosive or you needed to be fast, right? That's why you're more reliant on that system. Anyway, so you have these two, your 2 NAD plus became 2 NADH plus. You need it to become this again, right? So here's what you do. You take these 2-pyruvate, right? So what you're going to see later is we'll look at this process. You take a pyruvate, you take an NADH and an H plus, and an enzyme called lactate dehydrogenase that I'll talk about later, or LD, right, or LDH if you want, and you will make it into lactate. Now when you do that, right, so you can see here we had 2 NADH plus A plus. I kind of show them matriculating down here, right, 2 NADH plus H plus. What we do now is boop, boop, boop. Now these have become again 2 NAD plus. They can go back up here and be used in this process, and this can happen for a while, right? Like I said, if you're going at maximal rates, at near maximal intensities, kind of, Your glycolytic system can be your predominant system of energy from anywhere between like as early as some people 8 seconds, right? I would say maybe to like 50, 60 seconds to 1 minute into exercise. But you have to have this creation of lactate for it to do that, for you to have sufficient NAD plus for this process to go relatively unabated, unhindered. So the creation of it, right, is what really allows this to happen as rapidly as it does, because otherwise you're going to have to rely on the mitochondria. It's an efficient system, but it's a slower system. You can't make ATP as fast. So, very cool stuff. So what I'm going to do, I'm going to flip the board, and we'll go on to our next point. All right, so I'm back with part C. As far as our second myth about lactate, right, our myth that lactate is simply a waste product. So what we're going to look at now is how you can actually take lactate, turn it into glucose, and get it back to a muscle that's working. Right? I'm going to do a really simple presentation on it. It's disgraceful. I'll go more into all this in depth when I go into in-depth work, but to make this presentation as simple as possible, that as many people as possible can kind of grasp onto it, I'm going to keep it simple. For those of you who understand these processes, you'll be able to look these up and go into much more in-depth into it. So there's a process. It's named the Cori cycle, right? Eponymously named for these couple of, some, I think, Austrian and something else American scientists. This married couple and then this Argentinian scientist. Anyway, they won the Nobel for it, so it's like a big deal. Anyway, so the Cori cycle, right? But what it really is it's a fancy way of what we say if it's creating glucose, right? It's a special way of creating glucose. So it's what's called gluconeogenesis. And so what I want to do is kind of break these words down so we kind of know where it's coming from, right? So gluco, right? And we can see that in glucose, right? So that's what it's talking about. Neo is going to mean new, and genesis is going to be creation, right? So we are creating a new glucose. Now, specifically though, you're creating a new glucose from a non-carbohydrate, right? So this is so that's what we call it, gluconeogenesis. Anyway, so in general, right, we've already kind of talked about this, but in your skeletal muscle, you're going to have glucose, probably in glycogen stores, and we didn't get all into that in glycogenolysis, but another time. But we're going to say at some point it's going to be glucose, right? You're going to take that glucose, you're going to turn it into 2-pyruvate, and then you're going to turn it into probably into lactate. Not all of them, but a lot of them, right? These are going to leave the muscle, right, through monocarboxyl transporters, get put into the blood. Now, one of the places I talk about is that it's going to go, right, that it's going to use it as fuel is liver. But that's not the only thing it's going to do with it. What it has the capacity to do is take this lactate, turn that lactate back into pyruvate, right? It kind of flips this process. We'll talk about that later. I'll go into it in depth, how that kind of happens. But for now, we're just going to say that it happens. You have to take my word for it. I mean, I've never seen it happen. I've only read about it in books and such, so I assume it happens in my body. The lactate becomes pyruvate. Then you take that pyruvate, you turn it into glucose. Then the liver dumps that glucose into the blood, right? A lot of your blood, when you're exercising, and you know this already intuitively, is going to your skeletal muscles, right? You have this kind of what we call like transient hypertrophy, right? Those areas that are working are typically swollen, like bigger than normal, right? Because there's so much blood flow going to those areas, right? Anyway, so when it gets into the blood, right, a lot of that glucose is going to go to these working muscles, and then the process repeats itself. Now, you're not going to be able to rely on this, let's say, as a source. I just want to be very clear. You can't, like, for every glucose spent, you don't get a glucose back, right? It doesn't really kind of work that way. It's not going to come back kind of that way or whatever. This can't work in perpetuity. So I just want to be kind of clear about that. But it's something that helps you keep glucose levels relatively stable, let's say, especially like within the blood and things like this. One of the mechanisms that you'll use to do that, right? To make sure that the skeletal muscle is getting its supply of glucose, but so is your brain, right? We'll talk about that kind of later on, but I'm hoping that kind of makes sense. We can follow that pretty well. I don't think it's overly complicated. The processes that I'm skipping right here are much more complicated than I'm going into. But I don't want to go into those processes now. For now, I think it's keeping it like this, just relatively simple is the way that this would go also. All right. So, anyway. There's no reason I just don't have my red marker with me. That should be red blood. All right. That's it on that. I'm going to flip the board.